Hello and welcome. My name is Amanda Grimm. I'm the Senior Segment Marketing Manager for Antibody Drug Discovery and In Vitro Diagnostics at GenScript. Today my talk is titled A Comprehensive Portfolio of Advanced Tools for Cancer Research and Therapeutic Development. The contents of this presentation include an introduction to GenScript, cancer and strategic targeting, solution areas of interest. I'll cover some publications of GenScript in the literature, and then I'll close out with a summary. For those of you not familiar with GenScript, we are the leading global synthetic biology company. We have offices around the world, including the UK, the Netherlands, Singapore, China, and Japan. We were founded in 2002 in Piscataway, New Jersey, here in the US, offering the first custom gene synthesis platform. Last year, we celebrated our 20-year anniversary, commemorating our growth as the GenScript Group, adding subsidiaries like Legend Biotech that specialize in cell therapy, Custom Array, which is our Seattle-based oligo and custom array production facility, and also our CDMO, GenScript ProBio. Also in the last few years, we've added our catalog products division, where you can find automated instruments and ready-to-use reagents. Let's go ahead and get started talking about a little bit about cancer and some of the strategic targeting involved. So I just want to start off with a couple of statistics related to cancer as the leading cause of death. These numbers come from the National Cancer Institute, which reported some global numbers in 2018 with 18.1 million newly diagnosed cases and 9.5 million cancer-related deaths. This really just emphasizes that the work that you're doing is really, really important in helping diagnose people with cancer as well as developing the treatments. As far as treatments available, historically, we know about chemotherapy and radiation, and more recently, we're seeing more along the lines of treatments for precision targeting. When we look at precision targets, we're looking at aspects of the immune system, the cell growth pathway, how the cells grow and divide, the blood vessel formation and growth, cell death due to substance or drug introduction. We're also looking at the cell death pathway as well as cell starvation. From those precision targets, I just want to narrow in a little bit more into the cancer immunotherapies. This is where the immune system is being harnessed in order to fight cancer. Data is showing this to be more precise, more personalized, and a more effective method than current cancer treatments like chemo and radiation. This is a graphic from the Cancer Research Institute just outlining an overview of those immune system players. So we're looking at the different T cells, B cells, antibodies, antigen-presenting cells, as well as cytokines. And then here we have a list of those specific therapies using the immune system. So looking at antibody drugs, cell therapies, gene therapies, and cancer vaccines. The first of these immunotherapies is the antibody drugs. The goal of antibody drug discovery is to develop safe and effective drugs that can selectively target and neutralize disease-causing agents while minimizing off-target effects. Here I have an illustration just giving an overview of the development process broken down into these 4 stages. The process involves the identification of target antigens, the selection of appropriate antibody formats, optimization of antibody-antigen interactions, and rigorous evaluation of safety and efficacy through preclinical and clinical trials. Taking a look back at the history of antibody therapy development, the first FDA-approved antibody was approved in 1986. There was a slow progression from that time until about 2006, and then after that, we can see that the FDA started approving new antibody drugs on an annual basis. And then when we get to 2021, the FDA has approved its 100th therapeutic antibody that I'm sure we're all familiar with, since this was the monoclonal therapy for SARS-CoV-2. Here I have a table of the top 10 FDA-approved monoclonal antibodies. The leading antibody drugs target cancer, and these can be seen highlighted in red. These include treatments for melanoma, colorectal cancer, and non-Hodgkin lymphoma. And then just to note, not too far behind cancer, we have therapies for autoimmune diseases, and these are shown in purple. So in this slide, I just want to highlight a couple of those antibody therapies and what exactly they're targeting. The first one up here at the top, we have Herceptin. This was developed by Genentech and was FDA approved in 1998 for breast cancer. It targets the HER2 receptor, and the HER2 gene makes the HER2 receptor that are on the surface of all cells. When a patient's HER2 positive, it means that these cancer cells are overproducing these receptors, thus able to grab more of the growth hormones and chemicals floating around, resulting in their uncontrolled growth. Blocking the receptors blocks the growth signals. And then another activity that Herceptin is able to do is to alert the immune system to destroy the cancer cells that it's attached to. The other therapeutic we have here on the bottom is Keytruda. This was developed by Merck and was approved in 2014 for melanoma. Keytruda is a checkpoint inhibitor where PD-1 is an immune checkpoint receptor that, when bound, turns off the T cell activation. Blocking PD-1 on the T cells from binding the PD-L1 on tumor cells. This now allows the T cells to become active and continue with the proper pathway to kill melanoma cells. The next immunotherapy is cell therapy. This is essentially using cells from a patient's own immune system to target and destroy cancer cells. An example of an FDA-approved therapeutic is Kymriah. This is used to treat certain types of aggressive B-cell lymphomas and acute lymphoblastic leukemia. Kymriah is a type of CAR T-cell therapy. This means that it involves removing the T cells from the patient blood, engineering those T cells to then recognize and attack cancer cells. Next, we have gene therapy. This involves modifying a patient's genes in order to help fight cancer. This can be done by replacing or repairing defective genes or introducing new genes. One example of an FDA-approved gene therapy is Luxturna, which is used to treat a rare form of inherited vision loss caused by mutations. Luxturna works by delivering a normal copy of the gene to the cells in the retina. This then restores their ability to produce the protein needed for vision. This is not specifically used to treat cancer, but it is an example of how gene therapy can be used to target specific genetic-based mutations and restore normal functions to the cells in the body. And then the last type of therapeutic that I mentioned is cancer vaccines. These have shown great promise in studies. Essentially, we have 5 vaccine types. We have mRNA, recombinant protein, DNA, viral vector, and peptide. Vaccines aim is to stimulate the immune system to target and destroy cancer cells. The vaccines themselves can be made from cancer cells or from substances that mimic cancer cells. Let's go ahead and take a look at our solutions of interest of where GenScript can help you with your cancer immunotherapy development. Here I have an overview of the solution areas I'll cover. This includes gene analysis, protein analysis, cell-based analysis, and I'll touch on some of the preclinical and clinical support that we can offer. Just want to mention that this is by no means a limited list. This is just what I've selected to cover in this presentation today. So starting with gene analysis and editing, we'll look at gene editing, some molecular biology, and mRNA. So we'll go ahead and start with the GenTitan 8 million oligos and NGS libraries. Oligo pools allow to screen a large number of genes at the same time. These are mixtures of oligonucleotides that can be designed to target specific genes or gene families. They can help knock down the expression of multiple genes in parallel, which will then help identify the genes that are involved in a particular biological process or disease state. They can also be used to perform genome-wide screening, allowing for you to identify novel targets or pathways that may not have been previously considered. They do offer a cost-effective and efficient option, especially when you do have that large number of genes that need to be tested. DNA synthesis is a powerful molecular biology tool that has replaced traditional cloning and mutagenesis techniques. Our sequence optimization platform ensures 100% accuracy and recombinant protein expression. I have the workflow shown here, looking at the DNA sequence input, followed by oligosynthesis, then gene assembly, cloning, and then plasmid prep. We can also cover different production and quality levels, and that starts with research, industrial, high throughput, all the way to preclinical. You could also add our mutagenesis service that would allow you to create specific mutations in the genes of interest. This could then be used to study the function of the gene, develop new drugs, or create novel proteins with desired properties. We offer several options for site-directed mutagenesis, including point mutation, deletion, and insertion. We offer different service levels, so depending on your need and the number of base pairs, we can get a project turned around in 8 to 10 days. Expanding our mutagenesis tools includes our precision mutant libraries. The libraries are created using site-directed mutagenesis to introduce a defined set of mutations into your target protein or antigen. This, for example, could then be used to screen for antibodies with improved binding affinity, specificity, and functional activity. Precision mutant libraries can be generated for various targets, including difficult to express proteins as well as membrane proteins. If you're working with PCR or qPCR in your research, or maybe you're working on developing a diagnostic assay, our PCR products include high-fidelity DNA polymerases, master mixes, and PCR cleanup kits. For qPCR products, that includes the qPCR master mixes, probes, and primers for various gene expression analysis applications. GenScript also offers gene synthesis services in order to produce custom DNA fragments for use as primers and probes. CRISPR has become popular due to its ease of use, high efficiency, and specificity. One approach for using CRISPR for non-viral therapy development involves harnessing the cell's natural DNA repair mechanisms, such as the homology-directed repair, or HDR, to introduce precise changes into the genome. HDR is a repair pathway that uses a template DNA strand to precisely repair a double-stranded break. in DNA. It can then be used in conjunction with CRISPR to correct genetic mutations or introduce new genes into the genome. Here we have GenScript's 3 products that can be customized to match specific genomic target sites. The first one is our GenExact single-stranded DNA. It's a single-stranded DNA molecule that can be used as a template for CRISPR gene editing. This is ideal for T-cell therapy, has minimal cytotoxicity, and has an insertion length of 150 to 5,000 nucleotides. The next one we have is our Gen1 double-stranded DNA, which, as the name implies, is double-stranded DNA molecule that can be used as a template as well. This would be ideal for long gene CRISPR knock-ins. It has the covalently closed ends, which is protection for better accuracy, and this would be ideal for insertion lengths from 2 to 10 kilobases. The third one is our GenCircle, which is the circular double-stranded DNA molecule. This is ideal replacement for plasmid HDR template. If you're looking to increase efficiency, we've seen an increase by up to 30%. And with this, you can look at an insertion length from 1 to 50 kilobases. All 3 of these products can be customized to match specific genomic target sites. And you'll also note the production levels that we're able to provide range from research to cGMP grade. Another valuable CRISPR tool are gRNA libraries. An example is using them as part of antibody drug discovery for target identification, functional validation, and identifying resistance mechanisms. They can enable genome-wide screening to identify potential targets and can be used to validate the function of a target and identify drug combinations to improve a drug's efficiency. At GenScript, we have pre-designed pathway libraries that have been validated by the Broad Institute that include, for example, at the top, you can see acute myeloid leukemia as well as Alzheimer's. And we can also fully customize a library for your specific project as well. mRNA has become a common household word due to the COVID-19 pandemic and the development of mRNA vaccines by Pfizer and Moderna. At GenScript, we offer a comprehensive mRNA synthesis service for those of you looking to study gene expression, develop new therapeutics, or create mRNA-based vaccines. One of the key challenges associated with mRNA synthesis is the inherent instability of mRNA molecules, which, as you know, can degrade quickly and lose function. To address this challenge, we use modified nucleosides and optimize synthesis conditions to improve the stability, which helps to increase their shelf life and improve their performance in downstream applications. In addition to the modified nucleosides and the codon optimization, we also add 5' capping and optimize the poly A tail. Another relevant RNA interest are circular RNAs, which are single strands of RNA that form a covalently closed loop. Previously, they were thought to be an aberrant splicing byproduct, but are now being explored as an application in mRNA therapeutics. On this slide, we can see the chart on the left from PubMed, reflecting the significant increase in publications related to circular RNA. That research is showing these protein encoding circular RNAs to have better stability for longer expression duration, higher protein expression, and better thermostability. Moving away from RNA and onto plasmids. If you're working on plasmid purification and looking to automate a MIDI-level purification, we recently launched our AMAG Quattro, which is a high-throughput purification system. It can process up to 24 samples in one run, with each module able to purify 6 samples each. It utilizes magnetic bead technology for efficient and rapid purification. You simply insert your samples with our prepackaged reagent consumables, and in 2 hours you have purification results with quantities between 300 micrograms to 1.5 mg per sample. The instrument control has an intuitive touchscreen interface that allows for easy operation and monitoring of the purification process, while each of the 4 modules can be run independently. Let's take a look at our protein analysis solutions. This will involve antibody development, protein expression, and purification and detection. Mouse hybridomas are the traditional method of generating monoclonal antibody. Our MonoExpress service provides multiple options to generate high-quality leads. Our process is made very simple, and all you have to do is provide us with your target sequence. We will express the protein, prepare it, and immunize the mice. Our MonoBoost immunization is an optimized technology that can help expedite your hybridoma project compared to the standard method, and this generally eliminates about 3 to 4 weeks of development time. We offer 2 additional methods of antibody lead generation using B-cell screening and cloning that improves the timelines that you achieve usable hits. Our MonoRab monoclonal antibody service uses the rabbit immune system to generate and isolate B cells post-immunization. Rabbits are known for their high affinity and avidity antibodies. The service line shown on the top is simple enrichment of antigen-positive B cells from isolated PBMCs, which is then followed by screening and harvest. To further expedite your timeline, we also offer the Beacon system in combination with the rabbit B cell generation. The Beacon allows for high throughput screening since it can test a high number of B cells and perform multiple screens in parallel. Screening that typically takes a week or more can be completed in a day. The high quality leads are then isolated and sequenced, followed by engineering for recombinant antibody production. We additionally offer protein expression support with multiple platform options based on your timeline and post-translational modification needs. For mammalian, we have CHO or HEK 293 cells. For bacterial, we use an E. coli system that is fast and very cost-effective. And we also offer an insect expression platform. As mentioned, our bacterial is the least expensive and very quick. If your protein isn't needy, this is a great option to get protein in approximately 3 to 4 weeks from sequence to purification, as shown down here on the bottom. To look more at our CHO options, the TurboCHO platform is a proprietary technology that utilizes high-performance CHO cell line to improve protein and antibody expression and yield. We have 3 different levels of service that are applicable to the different stages of drug discovery. Our high throughput, which is our small scale transient expression. We have Express, which is our high titer and can accommodate many antibody formats like IgG to bispecific. And then we have our high performance, which is used for scale up and getting more of those gram level quantities. The platform offers several advantages over traditional CHO cell lines, including higher expression levels, improved cell growth and viability, and reduced production costs. At our shortest timeline, we can have antibody to you as fast as 2 weeks. For in-house analysis, we also offer gels and blotting automation as fundamental tools for protein detection and analysis. This includes the blotting units, membrane processing instruments, electrophoresis standards, and high-quality antibodies for screening. From a personal standpoint, anytime you can automate Western work is a must. With the amount of time that you spend making gels, dealing with uneven transfer, and handling a million tubes that you might use for staining. GenScript also offers a variety of immune checkpoint proteins and antibodies, which are essential for studying the immune system and developing your immunotherapies. Immune checkpoints are regulatory molecules that help prevent overactivation of the immune system, but they can also be exploited by tumors in order to evade immune surveillance. By targeting these checkpoints, you can potentially enhance the immune response against cancer as well as other diseases. And our immune checkpoint products include recombinant proteins, antibodies, and ELISA kits. For your purified proteins and antibodies, GenScript offers a range of services related to antibody and protein labeling. This includes biotin and HRP conjugation or potentially adding a fluorophore. The labeled antibodies or proteins can then be used in a range of applications such as immunofluorescence, flow cytometry, or Western blotting. Our ELISA development service is designed to help you create custom ELISA assays for the detection and quantification of proteins of interest. Additionally, providing you with a method to investigate protein function, detect protein-protein interactions, and develop diagnostic assays. Options for ELISA development include standard ELISA kits like the sandwich ELISA, direct, indirect, or competitive ELISAs. We can also help with development for specialized ELISA kits related to pharmacokinetic needs, anti-drug antibody detection, or host cell protein. If you're looking for solutions for antibody or protein purification, our AMAG assay instrument can help automate that purification in-house. It'll save you time and labor while providing a closed environment to minimize endotoxin contamination. And you can purify up to 12 samples at a time, with that cycle time lasting about 50 to 60 minutes. Let's take a look at our cell-based analysis solutions that include cell line development, stable cell lines, and our cytosync cell isolation instrument. Cellular analysis is commonly used to clarify the target role in a signaling pathway or its ability to bind to ligands. Stable cell lines are a valuable tool in antibody drug target validation. They provide a consistent and reliable source of cells that express protein at a known level that enable you to evaluate the function and identify potential drug candidates that selectively target the protein. Our cell line solutions include a collection of ready-to-use stable cell lines. This includes hundreds of GPCR options, most of the immune checkpoints for cancer therapy development, various SARS-CoV-2 proteins, as well as the Claudin lines. And then, of course, if we don't have what you're looking for, we also have our custom cell line development. And that customized service is our cell power cell line development. Down here on the bottom, I have the workflow that outlines the entire development process and what is included at each stage, from vector construction, the cell line development, validation, and then delivery to you to start your experiments. Here we have GenScript cytosync cell isolation instrument. This is a tool to isolate single cells, small populations, or rare cell types from complex biological samples, such as circulating tumor cells or CTCs. It combines microfluidics and laser technology to capture and isolate cells based on their physical characteristics such as size, shape, and density. The instrument is also highly customizable, which allows you to tailor the isolation protocol to your specific research needs. This slide here gives you a sense of the ease of use for the workflow. Here at the beginning, number 1, you're labeling your cells with the beads. Second step, centrifuge to remove the excess beads. Number 3, run your cell sample through in order to isolate it. And then in the last step, number 4, you can analyze your recovered cells. This is our last section, looking at preclinical and clinical development. And within that, we have bioanalytical testing, preclinical production, and clinical development. The preclinical phase is a critical stage. This is going to help identify potential therapeutic candidates with the highest likelihood of success. The results from these studies provide valuable information about safety and efficacy. They help you refine the drug's formulation and manufacturing before it enters those human clinical trials. So there are several key steps, including in vitro studies that involves studies that evaluate the potential effects on cancer cells in a controlled environment. We also have in vivo studies that involve animal models such as mice or rats to evaluate the effects on living organisms. And then we have toxicity studies that are going to evaluate potential toxicity and side effects. And then lastly, we have formulation and manufacturing. In this slide, I've outlined the different levels of bioanalytical testing. So starting with bioassays, we can offer custom assay development to help evaluate drug activity and potency. We can also help to assess the immune checkpoints, provide cell viability, proliferation, and apoptosis. From the analytical side, we can help with methods to measure drug concentrations. We can help characterize your proteins, looking at molecular weights, charge, the N glycans. And then from the toxicology standpoint, we can help provide in vitro and in vivo studies in order to evaluate those toxicity and side effects. And then we also have endotoxin kits that can be used for detection and removal. My slide here summarizes many of the services discussed so far in the presentation, really highlighting how many of these we're able to offer according to production requirements that are aligned with preclinical production. So our antibody protein expression, our CRISPR products and surfaces, our cell isolation and activation, as well as our plasmid preparation. The final stage is clinical development. The goal here is to conduct clinical trials in people according to phases outlined by local regulatory bodies. In the US, the FDA outlines 3 clinical phases, where the first phase is a small group of patient and normal volunteers. The data is collected to assess dosing, side effects, drug metabolism, and pharmacogenic actions. In phase 2, we have a larger group of subjects and studying the effectiveness for the indication, identifying any short-term side effects, as well as risks. And then in phase 3, this is an even larger subject pool that is studied for the effectiveness and safety, evaluating risk-benefit, and determining basis of physician labeling. The clinical phase is complex, and it does require strict regulatory adherence. Our clinical development support is provided by our highly experienced CDMO team over at GenScript ProBio. They offer expertise in scale-up, tech transfer, as well as regulatory compliance. And then this chart is a compilation of our key services. Some of these were covered in today's presentation, like the antibody development, protein expression, CRISPR, and IVT mRNA. You can see that we're able to cover the majority of our solutions from early target identification to preclinical IND and into that GMP level manufacturer. You'll also notice that some of these are areas where we're working on adding GMP, and we do expect those very soon. Let's now take a look at some research publications that use some of GenScript's products and services. For the first publication, we're looking at work done by a group out of Thailand. Their goal was to generate high-potency chimeric anti-PD-1 monoclonal antibody using a highly efficient hybridoma generation and screening strategy. So over on the right, I've listed out the different GenScript services. And to start at the top, we have CHO K1 cells expressing human PD-1 that were used to immunize BALB/c mice for anti-human PD-1 antibody. Next, we have recombinant human PD-L1 Fc chimeric protein. This was used in screening assays for antibody selection. And then next we have them generating and producing chimeric anti-PD-1 monoclonal using the pcDNA 3.4 expression vector. And then this last one, they actually outsourced this mixed lymphocyte reaction assay to our GenScript scientists to perform, where with the assay we additionally measured levels of IL-2 and interferon gamma, and then compared the results against Keytruda and Opdivo as positive controls, and then had an IgG4 as the negative control. For our second publication example, we have a collaborative project by those listed in the bottom right. Their ultimate goal was to understand the structural basis of an immune response against human metapneumovirus and identify guidance for future HMPV antibody and vaccine development. On the right, like before, I have the GenScript products and services, starting with optimization and stable cell line development to subclone heavy and light chain variable region sequences from single-sorted human memory B cells into a PTT5 vector for expression in CHO3E7 cells. They also use our protein A column for antibody purification, and then also our ready-to-use strep tag for antibody labeling for serum absorption assay. And this brings us almost to the end. I've covered quite a bit, so let me run through the next couple slides just to summarize. The cancer therapeutic industry has seen significant growth and innovation in recent years. Antibody drugs, cell therapy, gene therapy, and vaccines have emerged as promising therapeutic approaches for cancer treatment. GenScript will be there to provide the tools and resources you need to make your cancer research easier through a range of products and solutions, of which I only covered a portion of what we provide. Areas of interest include gene synthesis and editing, CRISPR, IVT mRNA, antibody development, protein expression, automation instrumentation, and then I also covered a couple publications today that provided some examples of how different products or services can be used for unique project needs. And then here again is that overview of the solution areas, a visual outlining the gene analysis, protein analysis, cell-based analysis, and preclinical and clinical support that GenScript can provide. And with that, I just want to thank you for your time and learning the range of solutions that we have at GenScript. I'll leave my contact information here for you if you have any questions or if I can connect you with one of our specialists. I also have a QR code there if you want to head over to the webpage and learn more about these individually.